Imagine a vast library filled with instructions. This library holds the secrets to building a human being. This is our genome, our complete set of genetic instructions. It's written in the language of DNA. Our genome is breathtakingly complex, yet elegant in its simplicity. It's a story told in just 4 letters, A, T, C, and G. These letters represent the building blocks of DNA. They pair up in specific ways, forming the rungs of the DNA ladder. This genetic blueprint is organized into structures called chromosomes. We inherit these chromosomes from our parents. We have 23 pairs of chromosomes for a total of 46. One from each pair comes from our mother and the other from our father. Within these chromosomes lies the code that determines our traits. From the color of our eyes to our predisposition to certain diseases, it's all there. Unlocking the secrets of the human genome is like opening a treasure chest. We are only beginning to understand the vast amount of information it holds. The more we learn, the more we realize how much more there is to discover. Our cells contain 2 types of genomes, nuclear and mitochondrial. The nuclear genome resides within the nucleus of our cells. It's massive, containing over 20,000 genes. This genome is responsible for most of our traits. Think of it as the main library in our cellular city. The mitochondrial genome is much smaller and resides within our mitochondria. Mitochondria are the powerhouses of our cells. They generate the energy we need to survive. The mitochondrial genome has only 37 genes. These genes are primarily involved in energy production. Think of this genome as a specialized library within our cellular power plant. While both genomes are essential, they have distinct roles. The nuclear genome is like the blueprint for the entire city. The mitochondrial genome is like the manual for the power plant within that city. Our DNA contains the instructions for building proteins. Proteins are the workhorses of our cells. They carry out a wide range of functions. These functions include transporting molecules, catalyzing reactions, and providing structural support. Only a small portion of our DNA, about 1.5%, actually codes for proteins. The protein coding regions of our DNA are called genes. Each gene contains the instructions for building a specific protein. Think of a gene as a recipe in a cookbook. The sequence of DNA letters within a gene dictates the sequence of amino acids in a protein. Protein coding DNA is essential for life. It provides the instructions for the molecules that carry out all of our cellular functions. For a long time, scientists thought that the rest of our DNA, which doesn't code for proteins, was junk. But we now know that this junk DNA isn't junk at all. In fact, it plays a crucial role in regulating gene expression. Non-coding DNA refers to the vast portions of the genome that do not directly code for proteins. These non-coding regions were once considered junk DNA. However, we now know they play critical roles in regulating gene expression. These regions act like volume knobs and switches, controlling how much of a particular protein is made. There are different types of non-coding DNA, each with unique functions. Ribosomal RNA or rRNA and transfer RNA or tRNA are essential for protein synthesis. Regulatory RNAs such as microRNAs and long non-coding RNAs fine-tune gene expression. These RNAs can silence or enhance the production of specific proteins. Understanding the language of non-coding DNA is crucial. It helps us understand how genes are regulated and how this regulation can go awry in diseases like cancer.